Control deck, that is Hercules away.
Copy, Hercules away.
Control deck, that is Hercules, past the transom.
Copy, past the transom.
Control deck, that is tether, all out.
Copy, tether is deployed.
Control, back deck, that's Atalanta off the deck.
Control copies.
That's a little hot.
I'm gonna switch to manual iris. It might go a little dark for a sec. Switching Atalanta.
Hercules is dive.
Just gonna turn on the craft.
There's a lot of plankton in the water. Yeah, Rye, is that... At first I thought it was plankton, and now I'm worried it's mud from the last dive, or I don't know. What is that, all that?
Am I good to provide an audio slate? All right. This is an audio slate for dive H2081. UTC time is 114750. Mark.
Control, winch.
Control, go ahead.
We are all stop 50 meters. Are you ready for control?
We're ready for control.
Sent.
Yeah. You ready? Me going?
Couple of fish. There were some fish.
1,350 meters.
You guys, we moved the waypoint 1, right? So it's closer to the summit. We probably...
Yes, we moved waypoint 1 to the new
Right.
coordinates.
So
Well,
that's
what's that
probably
approximate
1,100
depth?
meters deep, something like that. Shallower.
I was like 100 meters deep. Oh, we're almost
No,
there.
no. 1,100.
So about 1,250, you think?
The updated dive plan says 1,050.
1,050.
1050.
Oh, are you doing that? Oh, you're way ahead. Ooh.
Did you get that, Jeff? 1,050?
Thank you.
Yep.
Yeah, thanks.
So we're just going to crab over there on the way down. That's the goal.
That is the
What
goal.
am I doing? Yeah. Yeah, I'm staying where I am. Roger. Can we just look at the other gauges? It's the top 3 that we're most interested in, yeah. Yep. Thank you. Great. Perfect, Alex. What? No, that's perfect.
Abstract gauges.
Oh, okay. Fast. Josh, how fast are you comfortable moving the ship to waypoint 1?
Ooh, that's a ways, eh?
Yeah.
Well, they're going to be moving sort of sideways, so, whatever. 0.5 is probably fine.
Okay.
They can manage that.
Okay.
Yeah. Yeah. Sounds good. Ooh, I'm gonna slow down.
We have to wait till we hit 200 meters to start moving. Okay.
I think we should start moving. It's a ways. Yeah.
Okay. We'll start moving now then. Bridge nav. Can we start moving half a knot, bearing 212? Yes, please.
Let's see. Hi, everyone. When everyone's good, could we do introductions?
Probably just another minute or 2.
Okay.
Thanks, Megan.
And
Yep, I'm doing it. Just takes a bit of time. I already
whenever-
put a couple inputs in.
whenever you have a moment, ROV,
You don't
upper
have to slow
and
down.
lower
don't have to
WCAs
slow
are
down.
still both off.
It's good. It's all fine. It's gonna be great. Sorry, what's that, Max?
The WCAs are both off.
Oh yeah, sorry. I was
No
forgot
worries.
about those things.
Those things.
Those silly things. Let me turn those on. It'll take about 45 seconds.
Copy.
I'm gonna go to dive special.
So the WCA camera is back for the dive, huh?
It's back.
Okay.
Yep.
Do we still have the Thurber camera on?
We still have the Thurber camera.
Okay.
And I will turn that on for you.
Thanks.
Network bottles on. Science. DSE. DSE is on.
Copy. Thank you.
Problem.
Oh. What's that?
Squid.
Oh, yeah.
Goodbye. Hello.
Oh, there it is.
Goodbye.
Hello. So much money.
Okay, Amanda, I bet you we can do intros now.
Awesome. I'm really excited. Ali and hello everybody. My name is Amanda Dedicatoria and I'm joining you here as your science communication fellow. I guess we could just start with the back. Could we start with Noelle this time?
Absolutely. Ali, everyone. Good evening. My name is Noelle. I am sitting in the data logger seat this watch, and I am the science coordinator for the Ocean Exploration Trust.
And not just that, she's a superstar athlete, as we've all learned today. You're amazing. And then how about you, Greta? My savior?
My savior. So, hello everyone, my name is Greta Sartori. I work as a researcher for the Palau International Coral Reef Center. I'm in Science C2 tonight, and I usually take care of the eDNA sampling or environmental DNA sampling for the Palau International Coral Reef Center.
With great joy. She helped me find my toothbrush and my toothpaste.
Where were they?
They were behind the mirror. I didn't know that mirrors had like boxes behind them. So she came up to me in the mess and she was like, Amanda, what color is your toothbrush? And I was like, blue and white. And she's like, I found it. And I was like, oh my goodness. I've been brushing every 2 hours since because I'm so happy. So thank you.
Yeah, right.
And Jeff?
My name is Jeff Drazen, and I'm a professor in the oceanography department at the University of Hawaii. I'm a deep sea ecologist, and I specialize in deep sea food webs and deep sea fishes. And I am the science watch lead for this watch.
And incredibly amazing. How about you, Menzel?
Good evening, everyone. Wilgabsingay Ali from Palau. My name is Menzel Ngirmeril. I work for the Office of the President here in Palau, but on board I'll be the cultural liaison. And shout out to Brian. The word of the day is ongalagala, meaning to joke.
Woo! Awesome. Thank you so much. And how about you, Max, in your lovely yellow shirt?
Yes. My name is Max. I'm sitting in the video seat today, and I'm a video intern on Nautilus.
Thank you.
And my name is Ray, and I'm in the Atalanta seat.
Hi everybody, Josh Chernov. I'm the RV Operations Manager for OET and in the Herc seat today.
Hello, my name is Dan Torriello. I'm a Research Project Manager at the Center for Coastal and Ocean Mapping at the University of New Hampshire during the day, but tonight I will be your navigator.
Awesome. Thank you so much. How's everyone feeling today? Well, tonight.
Excited.
Yeah, what are you excited about, Greta?
I don't know, the shape of this seamount is promising.
What kind of shape is it? Like a rectangle? Are we talking about a square?
It's a C.
A C.
Isn't it?
A C-shaped seamount?
Yes.
It's a volcano with a crater in the top, but it looks like one side of the crater has failed and slumped away, and so it forms kind of a C-shaped ridge. And then there's a slightly shallower portion still in the middle with a couple of bumps in it that could be old lava domes or something like that. We have no idea. And yeah, it's a pretty interesting feature. It's very obviously an old volcano.
That is so cool. What do you think we'll find? This is such an unusual and interesting shape.
Well, I imagine, I'm not really sure what we're going to find in the middle. These little bumps in the middle, it just depends on how the currents interact with them as to what we see. But I'm imagining up on the rim of the crater, it's just going to be quite amazing coral and sponge forest.
Wow. I'm super excited. Menzel, do you know anything about like sort of volcanic activity in sort of like Palauan history? Anything that comes to mind?
Honestly, the only thing that I could, in terms of like folklore and stuff like that, I think the old people who first came to Palau, I think they had a limited understanding of geology, so they turned those volcanic activity into like legendary folklore. So that's culturally that's the understanding of it, but there's not that much that are could be related to our modern understanding of geology today.
Thank you for that insight.
Do we know what the recovery time is for this dive?
Right now it's still planned at 8 a.m.
Thank you.
Yeah. So what's the max depth that we're kind of expecting for tonight?
We're going to land at approximately 1,100 meters depth and work our way up slope to the end of the caldera of this volcano. And at the very summit, on the edges, the C shape that Greta was talking about will be about 750 meters.
Mm-hmm. Awesome. That's really exciting.
Hey, Dan, are you able to figure out how long it would take the ship to get to waypoint 1 at this speed?
We're going to hit the seafloor before we get there, huh?
Yep, we will.
Yeah.
Perfect.
Well, depending on the depth, I guess. It'll be pretty close. They could try and increase speed to 0.7 maybe.
So to our pilots, as we are descending to our waypoint, what factors are you considering to get there?
I'm just looking at our speed to the seafloor, and then we changed our waypoint to, just because of the delay, we wanted to move up a little further. So we're headed to waypoint 1 right now, which is going to take roughly about the same time it's going to take us to hit the seafloor, but we want to be stationary when the vehicles get to the seafloor. So the ship's stationary anyway. So we're just kind of taking that into consideration. So we decided to speed the ship up a little bit, and hopefully we can get there before the vehicles hit the bottom.
Awesome. Thank you so much.
Okay, I'm going to turn the craft hydraulics off. No.
There was a very long
Yeah, siphonophore.
siphonophore, yeah.
Oh, cool.
There goes a little fish. There's a Tina 4.
Wow. So, Jeff, I have a question for you.
Sure.
So I read that, like, at one point during your studies, you took a break to backpack around the world. Can you tell us about that? How'd you do that?
Sure. My wife and I saved our money for quite a few years. Actually, when we got married, we only asked for money for our trip. And after I got my PhD, we put all our stuff in storage, which was not very much, and got a couple of backpacks and traveled around the world for a whole year. We hit every continent except Greenland. And we even got Antarctica. It was awesome.
Wow. What were some of the highlights?
Oh, wow. It was a whole year. Highlights included sitting in a natural hot spring in the Antarctic on Deception Island, and then jumping in the -2 degree water.
Yo.
That was great. Let's see. We hiked the Inca Trail to Machu Picchu. That was fantastic. Just driving around in a beat-up old camper van in Australia and seeing incredible wildlife. Fishing up in the Northern Territories, actually skipping fishing lures off the top of crocodiles' heads.
Whoa, that's fun.
Yeah.
Was it planned, or did it just go by ear? Like,
we had not very many plans.
Nice.
We had a few plane tickets. We changed plans as we were going. Yeah.
Yeah, that's the way to go.
It was really fun. So anytime there was just something cool, we would just stop and spend some more time.
How old were you then? Or how young were you then?
You have to really backtrack. That's it.
I'm sorry.
Mansell.
I'm 29. I don't know what you're talking about.
He's only 2 years older than I am. Okay?
You don't have to answer that.
But if you want to.
Young. It was great. It was an amazing time.
Yeah, well, what kind of prompted you to do that? Was there a discussion like, hey, do you want to travel after this? Yeah. Where do you want to go? Yeah. Like.
Yeah, I mean, I just wanted to see the world, and I figured that the best time to do that was when my wife and I didn't have any other commitments. So, yeah, we planned it for. I mean, we didn't plan like the day-to-day stuff, but we planned on doing it and tried to figure out how we would save enough money to do it for a couple of years. So, but yeah, we just wanted to see the world. Just wanted to see all the nature everywhere and meet new people and go to new cities, and it was marvelous.
How would you say it changed you and your perspective as a person?
Oh, it really, really broadened my perspective for sure. I think there's so many people who don't really have the opportunity necessarily to travel about the world, and so you kind of think that most of the world must be sort of like what you experience in your home, and that's just really not true. There's so much diversity in culture and ways of living, and so much diversity of ecosystems and organisms, and it's, yeah, knowing a bit of what all of that is just really helpful in understanding the world. So yeah, I think some of the things that were really shocking was like seeing slums in Bolivia, and you're driving out of La Paz on the train, and you just go through, you're way up in the Andes. It gets really cold at night, and there's just miles upon miles of people living in tin and cardboard houses and things like that. That's really, really hard to see. But there's also just many, many amazing, amazing things. So, and meeting great people. We, in Mauritius, I saw some people fishing off the rocks, so of course I had to go over and talk to them, find out what they were doing. And it was a group of guys that all worked together, and they just happened to be taking sort of an afternoon picnic, and we started talking about fish, except one of them really mostly spoke French, but fortunately my wife is French, so she came over and started speaking to them, but before we knew it, we were being invited around the island and into their homes, and we got this whole personalized tour of this little tiny island in the middle of the Indian Ocean. It was great.
That's amazing.
Yeah.
You never knew what you would find on your adventure.
Nope.
That's amazing. Thank you so much for sharing.
Jeff, I love that anecdote because I always get made fun of for always having to ask fishermen what they're fishing for and what they're catching, but now I can tell that story. There's good reason I'm doing it.
You guys would be very popular here in Palau, actually.
Yeah. Tell us more, Max.
If I see someone fishing, I will just come up to them, as Jeff, it sounds like, does.
Yep.
And ask,
Always.
what are you fishing for?
What
And
kind of
are you catching anything?
What kind of reactions have you gotten?
Usually people are like, oh, I'm catching such and such a fish here. Are you catching anything? Some fishermen are very like, they guard their spots.
They're like, what are you,
Yep,
cop?
exactly.
Yeah. So some people might be reluctant to tell you how the fishing is, although they will tell you what they're fishing for.
That's interesting. Would you say that you've had like similar experiences?
I definitely guard my good fishing spots.
Yeah.
Exactly.
Thank you so much for sharing.
Uh-huh.
Noel, as I mentioned before, that you are our star athlete in the room. Could you talk to us a little more about that and your amazing story?
What would you like to know?
Just kind of what made you say yes to being asked, would you like to row across the Atlantic Ocean without ever knowing how to row?
I think that knowing how to row is maybe the the best skill that could be learned for that. Like we had all of the other experiences that were required and expertise to like navigate our way across the ocean and deal with remote settings and rebuild things that broke. So I think that learning how to row is kind of the smallest battle to wage in the training process, I suppose. But it was a very easy yes. It wasn't really something that took me a long time to decide that I wanted to do. I wanted to do it with those people, and it seemed like a good opportunity.
What were some moments during your journey in which you felt grateful that you had said yes to the opportunity?
Every sunrise. That was great.
Yeah. Thank you. Thank you so much for sharing.
Yeah.
I think a lot of us out here are very much interested in exploring one way or another.
Yeah.
And just seeing what there is in the world and going to places and seeing things that no one has ever done before. And that's, yeah. So sometimes it's hard to divide the personal life from the professional one because we're seeking the same sorts of things in both.
It's really interesting. I was actually having this conversation with Kim the other day. She kind of turned to me and said, ordinary people don't do this. They go to the grocery store, they visit their mom, they go to work, and that's kind of like their day. And I was just like, well, I guess that means that we're also extraordinary people. And ordinary is just kind of our part-time.
As long as you're content with what you do,
Yep.
your life is going to be extraordinary. That's what I think.
I agree.
But yeah. I don't know. I think it's really special to get to share a space with people who are just excited about life as you are.
Thank you, Amanda.
Yeah. Of course.
Greta, did you take a look at the currents in this area?
Right now? No, I didn't. Should I?
They're kind of crazy.
That's probably the same. Yeah, it's exactly the same as it was a couple of days back.
Yeah.
Yeah.
Big, huge eddy to the west, and just north of Toby here, it's kind of a little bit of a lull in the currents.
Yeah.
Yeah.
How is the current looking, Dan?
It was 2 knots earlier.
Stand by. We're seeing a surface current around a knot.
Oh, okay. Not as bad as it was earlier.
Oh, Dan, I just wanted to let you know and to thank you for letting me do the... What was it that you made me do?
What was it I made you do?
Well, we went to the back of the ship. You had a whole contraption.
Oh, yeah.
She let me...
We did an XPT cast to measure the speed of sound through the water column.
And what was the purpose?
Understanding how the speed of sound changes as you travel down to the seafloor is really important in being able to correctly determine the depth using the sonar system. Because the way the sonar works is we know the time that we've emitted a ping, and we know the time that we hear the echo. And so we need to understand the speed that the sound is traveling at in order to calculate the depth.
Interesting.
Dan, I have to admit, when I did it with you the other day, I thought it was going to be more exciting than it was going to.
Oh, I don't know about you. I was pumped.
I mean, it's pretty interesting, the science behind it, but I thought I was going to shoot something into the water.
Like
Just kind of falls off the end of the launcher, doesn't it?
Yeah.
Yeah.
Like, bloop.
all Dan asked was, do you want to launch this thing with me? And I was out the door already before he even finished asking the question. It was so fun just to see it plop into the water. I don't know, exhilarating. And then he was telling me, as you release it, make sure that you don't move so that the copper wire doesn't touch the ship. And as I released it, the first thing I did was move, and the copper wire almost touched the ship.
What happens in that case?
I think it, like, the system doesn't really read the findings as well. Thankfully, in that moment, it did not touch the ship. So I was just like super excited.
Yeah, you're transmitting a voltage up a copper wire from the probe back to the gun-shaped launching device, and so if you touch that copper wire to the ship is made of steel, so it's conductive, and so you bias your reading, and it makes the data not useful. But Amanda salvaged it. She didn't. You came close, but not too close.
I was just so excited. But thank you for that experience. That was super fun.
I'm glad you had a good time.
But yeah, did you manage to get a good reading today?
We did. We had to do a little bit of troubleshooting, and we realized that we had a faulty cable that we're going to need to re-terminate, but we were able to use a backup launcher, and everything worked out.
Yeah, I'm glad it was resolved. Thank you so much for that experience again.
Thank you for your help.
See, of course.
About 5 minutes to waypoint 1 now.
Okay.
Josh, we're close enough now. Wherever we touch down is probably okay.
Okay.
Oh, look at that eel.
Pretty.
That's a cool one.
Well, maybe why don't we stop the ship, and that'll allow us time to swing and catch up.
Sure.
maybe we'll let's stop like 50 off this time, because as you swing, it's going to get shallower.
Another siphonophore there.
Wow.
Okay. Well, our delta's pretty squared away, and I'm showing 60 meters off right now.
I feel like it's a bit less cold than usual tonight.
Yeah, it's not bad today.
That's probably good right there. Yeah.
How many layers are you wearing, Greta?
5.
5.
I still haven't put on my 6th one.
That's pretty warm.
Right? And I'm not suffering, so... Max, are you sure it's all good with the cooling system?
Yeah, everything is looking great right now, actually, so...
Wow. It's going to be a comfortable dive.
Greta, you better knock on wood or
I
something.
got a lock on there, too.
Yeah, find
We
some
can
wood,
do whenever
Greta.
you're ready.
You say that, but you still have how many layers on?
Just 5. Maybe because I have my merino wool this time.
Is that Italian wool?
I don't know. No, I don't.
Yeah.
Maybe. I actually don't know.
Sure. Yeah, you can come up a bit if you want.
Manzel, have you been thinking some names for these seamounts?
So the discussion that I had with the traditional leaders, they want to, so they're going to divide the entire EEZ to 16 to all the 16 states, and the traditional leaders are going to name them according to their own traditional protocols. So I recommended to have a prefix to all of them, and the prefix is Menzel.
Yeah. Fair enough, fair enough. I mean,
I'm just kidding.
you were the explorer, so.
They have
I'm
to
just
put your
kidding.
picture next to every name.
Maybe I'll give one to Brian. He really tried with the word of the day today.
Can you repeat the word of the day?
Ongalakal. And it means to joke or to kid. Ongalakal.
You did great.
Yeah, he
He was a
did,
trooper.
actually. If you guys want to see him say ongalakal, check out our Instagram page. It's on the story right now.
You shouldn't really miss out on that.
Yeah, it's riveting, actually.
Have we done fish yet?
Okay,
For
I'm
the
gonna see if
Palawan
I
word
can
of the day?
Spin around.
They did it on the last voyage.
I can see if I can spin around.
Sorry.
There's the seafloor.
Here we go.
Okay.
Grit, are you going to want a seafloor
Yeah, keep coming up. a
sample
little
and
more,
then also one 75 meters up when we hit...
I think maybe it's gonna be just 1,000 because
Okay.
basically it's just like in the sand, the 74 meters is not
Okay.
gonna be very significant, I think.
There are more fish in the sand than there are in the rocks.
You're right, Jeff. But yeah, I would say 1,000, and then so we have two extra new skins that we can trigger if we
Okay.
see something interesting. And I'm sure we will, with the summit at 750 meters.
Yeah, there's going to be some pretty cool stuff up there. It's a big pit there on the seafloor.
So Harold was telling me something very interesting. So apparently beaked whales
Yeah,
interact with the seafloor.
isn't
and
that
leave
crazy?
Marks there.
Yep, big, kind of curved, very long scours. This is hypothesized. No one's ever actually seen a beaked whale make one of them, but it's a reasonable hypothesis.
What does it look like again?
It looks like a trough, kind of like a crescent moon because they usually curve, and they'll be 3 meters long. And we've seen these on the seafloor in the Clarion-Clipperton zone over in the eastern Pacific, and people have seen them a couple of other places now too.
Cool.
They're often at depths of 3,000, 4,000 meters. Beaked whales are definitely the deep divers of the air breathers.
And what make people hypothesize that that could be due to beaked whales and not something else?
There's nothing else at those depths. You're below the depth of sharks and skates and things like that, and none of the fishes down there are big enough. No one can figure out what else would be big enough to make such a mark on the seafloor.
Some very well-motivated shrimp.
Huh. Well, we've certainly seen that with the tiny little trenches, haven't we? Well, they're not that tiny, actually. They're several feet long and an inch wide.
So actually, I remember one of our viewers actually commented about one of those shrimp canyons that we were looking at, and they were like saying about a beaked whale, and I didn't understand until now you were talking about it. I remember that comment.
Different scale, but yeah, there's been actually quite a lot of attention paid to trying to understand the various marks, trails, burrows, etc. on the seafloor over many years, really, and since people really started taking imagery from the deep sea floor back probably in the 1960s and 70s.
Interesting.
Yeah, it makes sense. It's just like tracking in the forest or something like that, trying to understand the signs of all the organisms that live there.
So are lasers off?
Lasers off.
Okay. Just give me one moment. There's some checks. All right. Okay, gonna go dark for a second.
Okay.
Okay, and then we're gonna go white balance. Okay, I think we should be good to go.
Okay. Yep. They look happy. Okay. Okay. I think if you're happy, Rye, you good? Okay, Jeff, I think we're ready to start.
All right, let's start making our way uphill towards, well, I guess waypoint 1 first, and then we're almost there.
Almost there.
Yeah.
Bridge nav. We go 30 meters, bearing 214.
So Josh, we will stop. It'll be a little bit, but we'll
Thank
stop at 1,000 meters to Tripaniskin.
Thank
Okay.
you, Jeff.
Sounds good.
Let's go check out this Calyptra right over here.
Yeah, point 3 is good. Okay, we'll zoom in, please.
Do we have any more zoom?
Absolutely.
Yeah, we can go in.
Sorry, Nav, what's the heading for this move? Okay.
That's great, thanks.
Okay, come wide.
Yeah, if we can just zoom in on that guy right there.
Okay. Sure, you got that right?
Yeah.
Sorry, can you hit me with
Yep,
one more little?
right
yeah,
there.
Okay, thank you.
Okay, Norbit is on.
And we'll zoom in, please.
And then after this, we should get more oriented towards uphill.
Okay.
Looks like maybe Norella. Okay, that's great. If we have a little bit more zoom,
Sure.
push in a little bit more on those polyps, and then we're probably good. Yep, they're all drooping down. Okay, thanks.
Okay, come wide, please. Sorry, bearing was, I know we just said it,
215.
215.
Yeah.
Just gonna have a quick peek on top of this.
Oh, cool.
Crinoids.
Some nice crinoids.
There's,
Oh, can we zoom on this Chrysogorgid over here?
Sure. We'll zoom in, please.
Is this just, oh, it's just an Iridogorgia. Okay, we're just looking at it head on.
It's very tightly
Very cool.
spiraled.
It really is. Great, okay.
Come wide, please. All right, I'll slide over.
fish.
Oh yeah,
This deal?
left, little, oh, if we can take a look at that, that is very, very long. That is maybe not a cutthroat eel. We'll see here.
Okay, we'll zoom in, please.
Nope, it is. Snaphobranchid. Can we get any closer view of the head of this fish?
Yep.
It's been through the ringer though, jeez.
Yeah, it looks pretty beat up. It's got broken fin on the anal fin towards the back. It's got scars on its snout here.
Is it missing an eye?
I can't tell from this angle.
Wow.
But this,
something
wow,
to-
the fin on this eel goes way far forward. I'm just hoping it will turn his head a little to the side so I can see the jaws, which will at least let me put it into a subfamily. It's probably Snaphobranchus affinis.
All right, I think I'm gonna have to get
Okay,
moving on. Okay, well,
thanks.
come wide, please. All right, I'll scoot up hill.
It shakes a lot.
Yeah.
Shakes his head, shakes his tail.
The Snaphobranchus really frequently do that.
Just fix my camera for
Or they gape.
Yeah,
it's tilted up.
absolutely. You're turned way down.
Yeah, I'm turned down a little bit.
And you also gotta be extended.
Great,
There you
that's
go.
great, thank you.
Alright, my heading is about 217 right now.
Perfect.
Wasn't quite sure what that was. Fish in the top left. And it looks like a piece of wood over here as well, but let's look at the fish. Excuse me, the fish first. That looks like a halosaur.
zoom in, please.
Yep, this is a halosaur. Aldravandia, I think. That's great.
Okay,
Let's go take a look at the piece of wood.
There, you can zoom in.
Oh, wow. We may want to stop the ship and take a look at this for a second.
Okay, let's stop the ship.
Bridge nav, can we stop the ship?
There's been a desire amongst some of the scientists to actually collect a piece of this wood to try to inventory the community.
Can you come wide, please?
So if we could perhaps sit down next to this just for a few moments and sort of scan it with the cameras and take a good look at it, and then drop it in one of the bio boxes.
Okay.
What's on it?
I'm not sure yet.
That's so cool.
Okay, we can zoom in, please.
Wow.
Wow, lots of brittle stars, but I'm not really sure what these other... There's a scale worm down in the bottom. There's a little crab on the left-hand side, little squat lobster, Munidopsis. But I don't know what these little white projections are sticking out of it.
Any more zoom? No more.
We are fully zoomed.
Can you tilt the camera up a little bit so we can look along the length?
That's also a gastropod there.
Oh, yeah. Okay, so this is a fairly well colonized system. These whale fall communities are now being explored all around the world, and they're really fascinating. There's a number of animals that we probably can't see burrowed into the wood. They're called shipworms. And in the deep sea, there's different species being described fairly regularly now. Yep, there's another snail up on the top
There's
there.
like a tiny sea spider also.
Ooh, where?
Oh, where's the sea spider?
It's on the dark part of the wood, about in the middle of the
Oh,
spread.
yeah, there.
Oh,
I
wow.
feel like that's the right piece of wood to collect.
Yeah. So this has developed quite a unique community. The animals that live on wood also sometimes live at cold seeps and other environments where bacteria, bacterial mats and things will grow. And in this case, what we've got usually are shipworms that bore into the wood. They use gut symbionts to digest that wood, as termites would do. And then that creates a whole ecosystem around them. And there's a lot more energy supply because of that on this piece. Yep.
Another
There's
species
another.
of
Yep. There's at least.
gastropod?
Yeah, that looks like a limpet right there.
Limpet.
Uh-huh. A
Or a
type of
mono-
gastropod.
Yeah.
Yeah, and look at this little turret shell right there. The fuzzy stuff you see coming out of the holes, that's probably the droppings of those shipworms, which are then actually used as a food supply by all kinds of detritus feeders. But no one's ever described wood communities from around Palau in this part of the world. So it'll be very interesting to see if the animals we collect here will match up to others found off of Japan and Hawaii and a few other places nearby, well, relatively nearby, where wood communities have been described. I think off of Australia as well.
Thinking about May and
Oh, look at that.
Noel doing the subsamples for this one tonight. No, tomorrow morning.
I'm thinking the whole sample will be pickled en masse. Yeah. There are very likely hundreds of animals on this piece
Yes,
of
I
wood.
don't think that's going to be possible.
Yeah. That's really great, Josh. Let's just slowly tilt up across it one more time,
Absolutely.
and then we need to figure out which bio box it will fit in.
Well, probably our biggest one, which is starboard, one of the starboard ones. It might not, well, we'll see. I hope it
So
fits.
Josh, yeah, I hope it fits too. It is possible that when we pick it up and try to put it in the bio box, it's going to break as well because of all the burrowing of the shipworms.
Yeah. Well, I'll try and be as delicate as I can.
It is okay if it breaks, because what folks will probably do when they check out the sample is they will break apart the wood to find all the organisms. So it's not the end of the world. If it falls in two when we start to pick it up, we'll just grab both pieces or a couple of pieces and put them in the box.
Okay. Sorry, I'm trying to do slow camera tilt up here. It's a little better.
There's some recent work on some of these woodfall systems off the coast of Oregon where they have a lot of wood, just as they do here because of the big forests. And in some of those experiments, they described several new species of little flatworms that were living in association with the wood, along with, I think, a new species of snail. So it's a really interesting system that adds a lot to the regional diversity. But it's really patchy. You only find it where you come across the woodfalls.
Yeah, it's going to be 063.
There's actually a second sea spider there as well, below.
Yeah, very small.
Brittle stars. They look like they're doing burpees or something.
Yeah. Down, up.
They're doing burpees.
Down, up. Down, up.
Josh, 1 second here. We're trying to get some focus issues worked out on the Thurber camera so we can get a couple of still shots.
Okay.
Yeah, I think you, Josh, are doing great. You're very still. It's not you.
It doesn't seem to want to focus.
You can select, so you should be able to select on the wood, like make a little box, little focus box. Is that, are you trying that?
So you go to manual focus?
Yeah, you should be able to select an area on the screen that it'll show focus on.
I know, I know. But
yeah.
I tried this many times before, and it's not getting in focus, no matter what I do. It feels like some days it doesn't really want to cooperate. Some days it works. So let's try the focus everything in focus. No. Like it goes in focus for a second and then it comes out. So yeah. You can go ahead, Josh. Oh, fish.
Oh. Bonus? Is it possible to zoom out a little bit? Oh, that's great. I love the shrimp. That's probably Plesiopenaeus right there, the shrimp.
Still there.
Tiny rat tail too.
Yeah, there's a little rat tail.
Or
oh, that's Bathygatus. Or a Bathygatid.
Ooh, hello.
Oh, wow. Excellent. Oh, I think it's trying to go into our net all by itself.
There's a huge fish right by the starboard bio box.
Another rat tail.
Yeah, I'm just getting you here so you can see. No, sorry, I should have warned you. You just have the settings very cinematic, so.
Hello.
Oh. Sorry, I was not on SPL. You've actually got a grip force setting. So, oh yeah, I would put it very light. Might drop things. Okay. Oh, nice. It's not gonna just fall apart. That's great. Oh, there goes one of the squat lobsters.
I was like, nope, not today.
Sorry, I didn't manage to keep you. Bonked a little bit. Just stick it right in this restaurant. Okay, box coming open. Okay. Maybe it will fit.
Oh, great.
Okay.
Outstanding.
Closed.
Which box did that make it in?
Big one.
Okay.
Yeah, I think that's F, maybe?
That is F.
Okay.
Can we get a quick zoom on the squat lobster after the arm is all settled?
Okay. Go for Sam. Your tilt up setting is really low. Like catastrophically low. Okay.
What is making this sediment mound?
There you go.
Not sure what that is.
Yeah, you
weird
can keep
thing.
going in. There we go. Let's have little orange eyes.
Yeah.
I like how it fell off and just went right back to business.
Gotta eat.
Just munching.
Okay. That is great.
Oh, that means it's somewhere. Yeah, that means it's somewhere. It's probably on yours though.
Ready to keep
Okay.
moving?
Yeah, we
Yeah, there
can
you go.
can zoom out.
Okay, zoom out. Tilt up is really low. Yeah. And you are back to your 19% on the PRV.
Some really dark sediment there that might have been a bit anoxic.
Yeah, maybe it's from the wood.
Yep.
Organic matter.
Yeah. Yeah, or it could have been organic matter coming out of the wood. Yeah. Wonderful. Okay. We can keep going.
Oh, bless you, Max. Are you okay? Do you need tissues?
No, I'm all right, but thank you.
No problem.
All this scouring around this
Oh,
rock.
Oh,
wow.
that's cool. It's like when
Can
you
we
go to
check
the
out
beach.
the sponges and corals here on the corner?
Here, I'll give you more tether.
There's some of these corals, it seems like the same we collected from the last seamount.
Oh god, I can't remember the. Yeah, it is from the same. The last seamount. I'm trying to remember the name of it. Acantha.
Acanthoprimnoa?
Thank you.
Oh.
That's it. Acanthoprimnoa.
Oh, look at that purple. Is that Victor
Victorigorgia. Yep. Got a bunch of zoanthids on an old sponge stock.
What's the small yellow? Is that
The
a coral?
sponge? I saw a demo sponge to the left. Oh, yeah, right up there next to the Acanthoprimnoa. That one? That little yellow one right up there?
I'm gonna
Yeah, I
close
think there's
the
a couple.
forward bio
Oh, and
box.
one of
Maybe
those.
Thurbicam will have an easier time focusing. Yeah.
Josh, is it possible to go just a touch higher? There's a little yellow coral right at the top center of the screen. Right there. That might be Enelopsamia. Do we have any additional zoom?
We have a little bit more zoom.
What is that?
Looks like brittle star
Brittle star.
arms sticking
Oh.
out. Yep, I think the yellow is Enelopsamia. I'm looking at it at a bad angle. I'm not certain.
Asako is typing.
Yes. Sako maybe can confirm some of these coral IDs for us. For those of you joining us online, we have a number of marvelous scientists on a special science chat who can help us identify and work through the information gathered on the dive. And right now, We have Asako Matsumoto from Chiba University in Japan. And Martina, I'm going to mispronounce her last name.
She's okay with it, she says. It's Gagliotti.
There you go, from an Italian.
Yeah, to an Italian. Fish.
Yep. I think
Well.
we're going to have to move. Yep.
Fish?
I'm not sure what that was. It might have been the umbellula, the little sea pen-like octocoral that comes up. Yep.
Should we try to get to waypoint 2, at least?
Ooh.
Oh.
Let's first take a look at that very large brittle star, though.
Forgot to put on sunscreen.
We've seen a lot of very small white brittle stars on this expedition, but not very many of these intense pink ones. If we have any more zoom, it'd be great to zoom in on the disk. That's great. Oh, that's really good. Okay. Thank you.
So are these brittle stars also living just in the sediment?
Yep. Many, many free-living species that live on or sometimes within the sediments. They're just so incredibly, the group is so incredibly diverse. And then you've got those taxa that are living, what we call snake stars, a type of ophiuroid that lives in the coral's arms. Many different species on sponges. It's an incredible diversity of that group in the deep sea.
Yeah.
In shallow water, too.
Yeah, yeah. I remember I was always playing with them very wrongly, but when I was a kid
Yeah.
in Egypt, they were everywhere.
Intertidal. Look at
Yes.
this trench right here. Isn't that interesting? What would make that?
Beaked waves.
Yeah.
Oh, some big rocks down there.
It is going to get very steep coming up here, Josh, so we might want smaller moves. Oh,
Oreo,
can we
I
just,
think.
is an Oreo? Okay.
Got that.
They're
that's-
always in the
that's-
steepest terrain. Oh, wow,
Yeah,
look on the
let's move,
right.
let's
Look at
move.
all the corals on the right. We may want to stop and check this out. Greta,
Wow.
or Noelle, can
Yeah.
you take a look at that image of the stone sponge
Yes.
Yeah.
so we can just kind of
There
put
you
our
go.
search images? We have a scientist from ashore who's interested in an unusual sponge that we saw on the last dive. We're hoping we might see it again this dive and be able to collect it this time.
Noelle had a very good look in the benthic ID, and she spotted some that are very, very similar, a bit better quality pictures as well.
Okay, okay.
So we sort of have an idea of what they look now.
Oh,
Yeah.
that's very cool. Let's look at that sponge right there.
Look at
Yeah, we're getting
Wow.
there.
That's a sponge?
That is a sponge.
No.
It looks like a ghost.
Isn't that amazing with all the different openings on the side?
What kind of sponge is this?
So cool.
I don't know, but I'm trying to find it out right now. Wow, this boulder is just covered in very
This is
small
cool.
things. That's a little
What is
poislastrid
that?
sponge there. The kind of chunky looking one. There's a bunch of them here. Big Iridogorgia, the spiral one on the left.
this rock with all these pinnacles.
Okay, I've got some stuff about 10 meters away from
Oh,
me.
wow.
I know we're stopped, but I'm still
Is there-
swimming a bit.
Possibility of checking out the whip on the right? So my best guess on the sponge is it was a Faria, but I'm not sure which variety. There's a number of different species, but I'm not sure about that.
The variability in sponge morphology has just been blowing my mind.
It's nuts. You can have two different sponges in the same genus and they look nothing alike.
Wild.
If we can zoom in on the very top of the whip, see the polyps there. That looks like it's been broken off. Well, maybe not. That's great. Thank you. I would love to take a look at that sponge, which might be the same as the wall that we saw kind of at the top middle of the frame.
You can see also the landslide from
Yeah.
all the rocks
Yep.
and how they are positioned.
Little mysid swimming in the field of view there.
Oh, rat tail.
Oh yeah, up in the water column.
Yep.
I'll go check him out in a minute. Him or her. What have
What
we
is
got
that?
here? I don't know. The right on the right is the sponge, and a lot of the brown stuff is also dead sponge, but this very segmented...
And is that a brachiopod that we see on the right, Max?
There is. There's a brachiopod.
Yeah. Whoa.
This may just be some sort of primnoid with a lot of its polyps closed up, growing in here. Snail back there. Yep.
Do you think this is the same sponge species?
I
feel like it looks a little bit
It
different.
looks different. Yeah, it's
Doesn't
not.
have the big
Doesn't have the
osculum,
hollow
yeah.
and all the oscula along the branches. It's something different. Not sure what it is.
Isako thinks the middle is Narella,
Okay.
That one.
with a question mark though. So.
Okay. Can we pan just a little to the right and zoom in? I don't know if we have any more zoom on
We are
the
full
on the
zoom.
white sponge. Great.
That is full zoom. You talking about that one?
Yeah. Great. Thanks. I think we got what we need.
Oh,
There
big
he
rat
is.
There's
tail.
the rat tail in the distance there. If there's any possibility we can go check out what that rat tail is. Maybe not. It may be headed over the side of the rocks there, so. Actually, this whip right here, I didn't even notice that before. If we're stationary and we can just zoom on that, that would be great.
What is that?
Well, I didn't see that at all.
Just a skeleton.
Okay. Let's zoom out. We may take a collection here. There is some interest in collecting some of this sponge kind of in the middle here. So let's take a small piece of that. This might be, well, I don't know, it's up to you, Josh, whether we want to put it in one of the boxes or clip it and slurp it up.
Sure, what are we doing? Okay. Thank you. Okay. We're doing a snip and slurp of the white sponge. Okay. Is crap valve on? Yep. Okay. There we go. Okay. Video. Can we go half zoom? It's pretty. Can we push in more? Yeah, tilt up a bit. Okay.
Yeah, kind of,
Yeah, that might be.
I don't
where
know,
can get
like
it.
snip there or something like that.
Yeah. See if I can get in here. That might be too low.
Oh, wow. Okay.
Here, I might be able to get oriented better. Can you tilt the camera down a little bit? Might be kind of funny. We'll see. Let's see if I can get this. Like,
Would it be better to come up? Yeah,
Yeah.
sure. I didn't realize that other one was so close to the rock.
Yeah. I'm just gonna get farther down here. Oh, there we are. Oh, I might have broke that off, so I'll grab that piece.
If it, okay.
What were you gonna say?
If it broke so easily, do you think maybe we should just slurp it from here? Do you think that if you try to grab it again, it'll break again? I don't know.
Probably. It'll probably snap into a few pieces when we grab it.
Okay.
Which, yeah, I guess could be an issue for slurping. Josh, what do you think? Yeah, I think it's broken off already.
Yeah.
Or at least partially.
It seems quite fragile.
Yeah.
I mean,
okay.
if we get a couple pieces with a slurp, it's probably okay.
Yeah, alright. Go wide. Sorry, beat me to it. I think I'm fine. I think I can get to the, yeah, it's fine.
Josh, I don't think you're on SPL.
That's by design.
Thank you.
There we go. Alright. Do we still have our jaw turned down? It's fine for this, but...
It's on grip force 1. Yeah, you might want to turn that up.
Nice, okay.
You can do it anytime.
Yeah, okay.
You can halt and then turn it up if you want to.
I think I'm fine. Just got it right now.
I gotta get the jar into the right spot.
Okay. Do a half zoom video. Maybe a little bit more. Yeah. Just want to see what I'm... Okay. And I'm good
Okay,
whenever
on
you're ready.
jar 1. I'll turn it up a little bit for you. And get the suction open.
Okay.
Oh, why does it keep doing that to me?
Oh, it keeps losing it.
Okay, yeah, I'll start there. So I'll turn the suction pump on.
Okay. See if we can just try to get
Suction's
what we
going.
were snapped off here. Yeah, that piece. Want to turn it up?
Yep.
I might be able to like.
Oh, there it goes. Part of it goes.
It's okay. Let her.
I could try to like poke it in.
Hold on a sec. Wait.
Okay.
All right, that's full suction.
That's full suction.
Yeah.
Okay.
Let me stop for a sec. And
Yeah.
Turn it down. And maybe just give it kind of a little tap on the rock, just the hose or something.
Are you trying to get it out or in?
In. It's in there now. I think it just needs a. There it
There
goes.
you go.
Yep.
Okay, go wide. And do we see it?
Uh-oh. Might be jammed in one of the bends there, huh?
Maybe, yeah.
If you back off, I could stretch the hose out a bit more.
Yeah.
Try to.
And turn it off for a second.
Yeah. Could also try reseating the suction tube. Sometimes that. Does it look like it's getting anything into the jar?
No.
Not really.
No. Maybe try suction off and then rotate. And then back in.
There's a little piece in there.
Yeah. There we go, that actually looks like it's doing something.
There's a small fragment in there.
Or maybe I can stretch out a bit. I feel like I just saw some.
Oh, okay, turn it
Did
off.
you get more of it?
Thanks, I just, little something came down. Yeah, okay, yeah, there it is. Yeah, it's in the jar.
Oh, good. There we go.
Score.
Yep,
Okay.
nice job.
All right.
Is that enough of a piece there?
I believe so, yeah.
It looks like it's nicely together,
So-
so I don't want to really turn on and whip it around the jar somewhere.
That's in jar number 1.
Number 1, yeah.
Uh-huh. And it looks like it's a Tritodictyidae sponge.
Okay, I'm back on
Okay.
flush jar.
Okay. That was the only sample we're taking for me.
Well, some of our experts would like to go back and sample the other one with the holes to our right. It's kind of down the side of the rock.
Could you point that one out for us?
It's not in the field of view.
Oh,
We'd have to
okay.
pick up and kind of just
Okay.
step over a couple meters and
Sure.
drop down.
Yep.
It's the very first sponge that we saw on the side of the wall.
Oh, okay.
Just a couple meters away from us.
Yep, right
yeah.
to that, yep.
Yep. Anthomastus, I think, the bright red one. Yep.
So it looks like we have 2 high schools tuning in from Georgia in the chat. We have Central Gwinnett and North Gwinnett High School. And one of them asked, how can you tell the difference between sponges and corals from so far away?
Well,
That's the one center screen there, sorry.
yes.
Yeah, okay.
I can say that definitely sometimes they trick you at a distance. As you get closer, sponges are usually white. That helps, but there are a number of white corals as well. A lot of the sponges really for a lot of the stuff you need to get close and zoom in. And then, of course, corals have polyps, and sponges are just tissue, usually with lots of little holes on the side, some varying sizes.
Oh, thank you.
This one What do you think? Might be just this, like, snip and put in a box, maybe?
I think so, yeah.
Give you a little bit more arm room here.
You're just gonna hover.
No, I'm going to try and perch against the wall, but I'm just going to turn on a bit of an angle so you can have a little more room to work with the arm.
Sounds good.
The rat tail made a guest appearance in the bottom right, so
Oh, I missed it.
still around.
Okay. Is it another one of those Kumba-looking rat tails?
It was large and very far away.
Okay.
That's all I can say.
Nice. Got a coral close up and upper too.
Yeah,
Can we zoom in on this one more time before we sample it?
Yeah.
absolutely. Okay, let's zoom in, please.
Wow. I'm uncertain if some of the frills on this thing, I think they must be part of the sponge, but it's really interesting how it has the tubes and then all the frills on the side. It's possible the frills are something else, little hydroid colonies or something, but okay. Yeah, let's sample this and put it in a box.
Which box do you want to put it in, Josh? Front box?
Asako
I
and
mean,
Martina getting very excited for this sponge.
Yep, this could be new.
Depending on the, I mean, I guess depending on the, yeah, I guess in this position we should put in the box, because I don't think we can even get to the hose right now.
Yeah.
Okay, we'll come wide, and I think we should go forward box.
Okay.
It's like a sponge imitating a coral.
Okay. Yeah.
It's really interesting.
Can we just stay full wide, please?
I feel like you find the sponges imitating
you.
corals, but not corals imitating sponges.
I'm just gonna check out the speed of the box.
How do you mean?
Yeah.
So you mean the sponges can be branched, but you don't ever see corals that are just a glob?
Yeah.
Shallow water ones.
I don't really see corals that remind me of sponges, but I see sponges that remind me of corals.
Yeah.
And you're gonna want to be retracted probably for.
Yeah, I'll retract when you're ready to do it.
What the?
Okay, you ready?
Yeah. Let's try this out. So, are we?
Love that.
I guess
You
probably
just-
all of it won't fit, right?
No, you just want to snip off, probably just the, I'm assuming you just want to snip kind of an outer sort of few lobes.
Yeah, something like that or something like that.
Yeah, okay.
I don't know. I mean, it's going to be hard. If it's not possible to just... Well, this thing I think is actually a little closer to us than we think it is, too.
Oh, it's right,
It is,
right
it's
in front as
very-
well.
Yeah,
close.
it may not be that big, in which case you may just have to snip it in the back. And
Yeah, I
Okay.
just gotta.
yeah.
Get then the sort of, just nudge the crinoid kind of out of the way.
Okay.
Gently.
Maybe we'll see it move.
Very
I've
gently.
been wanting to see one move, so. Alright, let's see if I can get this arm to do what I want.
Can deep sea crinoids swim
Yeah,
as
this
well?
is a bit tricky because
Like
you're
shallow water ones?
They do. Yep.
a little
Yeah.
cramped.
Not very often, but we have seen them do it.
Let's get this crinoid maybe. Up.
Maybe
Come
this
on,
time.
buddy.
Might be a second sample.
Or just like you, he wants to see
If
the
the
world.
crinoid gets sampled, it's not the end of the world.
Okay. I'm just trying to get oriented here.
You may want to come, like, from the top.
Think
You
so?
know what I mean? Maybe.
Or what if I...
Actually, that might work too. Put the shears like against the wall, like parallel to the wall, so like you rotate and then,
Yeah.
yeah.
And
Like, yeah, the
that
other
way.
Yeah, yeah.
Yeah.
And you just have to sneak the bottom jaw underneath.
Yeah. And just looking in upper.
Yeah, you got a little room.
Okay, I'm gonna open. Oh, sorry. Okay.
Do you want some zoom or?
Yeah, let's go. Just trying not to.
Uh-oh.
You got a little more room to the rock you see in upper.
Yeah. I kind of want to. Oh, sorry, I'm pushing you off. More like that. So I might end up taking.
Yeah, you're gonna have to go a little deeper, like all the way to the shear, right?
Yeah, I'm gonna go in like this. I think that's got.
Ooh.
Oh, it's very fragile.
Yep.
Yeah.
Okay, come wide, please. There, nice and slow.
Yeah.
That was
Okay, you want to
yep.
retract?
Yep. You can just hold there. Just wait
Okay.
for it.
Yeah, that's still draw setting 1, so.
Open.
Wonderful. Okay, I'm gonna just creep in here. Very.
Nice. Just try and come a little lower before you let it go. Let me hopefully drift on in there. And just try and do like a slow open if you can.
Yeah, okay.
And then just sort of hover the arm and let it fall. So
Yeah.
you use the arm to protect it.
Okay, that should be.
Yeah, that should be, that's great.
Okay.
Beautiful. Nice work.
Excellent.
Got everything off.
Thank you, guys. That's great.
Just lift up a little bit and I'll close the box.
Yeah, go for close.
Nicely done. That was awesome.
Okay, just gotta get myself back.
Just take a little look around while we're sitting here.
Yeah.
Is that another sponge there?
It is. That's a poisilastrid sponge.
Oh, okay, we can zoom in there. Let me look.
What kind of sponge is this?
It's a polychaete just underneath it.
Poisilastridae.
So we'll come wide.
Okay.
Is it possible that that is considered a stony sponge?
It doesn't look to me like these ones in here.
I agree.
What?
The best
Super
information
cool.
I can find was that
Yeah,
many
no, it
of
looks like
them,
you're in like a
the stony
diorama or
sponges,
something,
are
right?
in this
In that group?
order, maybe
Okay.
family.
Is that a homology?
But the stony sponges, from what I could tell, is not an official grouping. It's kind of a loose
okay.
term.
Okay.
So I'm not sure exactly what we're looking for with that.
Right.
Maybe we can get some more information from them.
Yeah.
But
These pictures, I believe, also came from very shallow on the last seamount, and so we may not see these until we get up another
Sure.
few hundred meters. I don't know that, but
Okay, are we ready to move
yep.
on?
Let's move on. Thanks very much.
Yeah. That was a really cool spot.
Bridge nav, go 30 meters, bearing 202.
Let's not do 30 meters. Let's do smaller steps, please.
Smaller, sure.
Yeah, because we're right up against the wall, right?
Okay.
So
Yeah, this is going to get even
You wanna
steeper
go like
up here.
10 meters?
let's do a 10-meter move first, and we'll kind of ease into it.
Bridge, scratch that. Let's go 10 meters, bearing 202.
Yeah, look at all this up here.
Wow.
Oop.
Big old crinoid. Oh, what is that floating in the water column to the left?
You're looking at that?
Yeah.
I don't know. We can zoom on it. That's probably a jelly. It is. And there it is.
We'll zoom in, please.
Oh, that's another one of those.
Yeah, it's another one
sponges.
of those sponges in the background there.
Wow, beautiful.
Oh, that's amazing. Hard
So
to focus
tiny.
on, yeah?
Yep. There we go.
Whoa.
Oh, there it goes.
Okay.
It's pretty.
Both of the different types of sponges here.
Are deep sea
Chrysid-
jellyfish different than the ones in the shallow, right? There are different group of jellyfish.
There are some different groups, yeah.
Yeah.
There's also a number, like there's a jellyfish paraphila that you can catch in fjords in Norway in 20 feet of water, and you can also see them at 2,000 meters in the tropics. So there are some,
There it is again.
and it's it's not entirely clear if they're exactly the same species. They may be. We've got some stone. Got a bunch of stony corals here, corallium.